Hi, my name is Yazi. I did my PhD here in University of Sydney. So I graduated with a PhD in neuroscience in 2009. And since then, I commenced my research career. So I did a lot of research in neurodegenerative diseases. And my expertise is on developing disease models and using these disease models and to understand what happens in disease. So that's a sort of brief background on my expertise and experience.
I'm Lars Ittner. I trained in medicine in Germany and Switzerland before I moved in 2005 to Australia to study the causes of neurodegenerative diseases. Over the past 10 years, I've built the Dementia Research Center as a hub for translating basic discoveries into novel therapies for neurodegenerative conditions such as Alzheimer's disease and ALS. I'm the Chief Medical Officer for Celosia Therapeutics, and my responsibility is to pave the way for the clinical trial of our lead asset, CTX-1000.
So I'm the Chief Operating Officer at Celosia Therapeutics, and my key role is looking at developing our current lead asset, which is CTX-1000, and moving it through the manufacturing phase, and then taking that CTX-1000 in its current form and conducting the different preclinical studies that's required to accelerate that towards first-in-human trials.
Celosia Therapeutics was formed around a portfolio of different genetic medicines for a range of neurodegenerative conditions, with its lead asset being CTX-1000 for ALS, which we hope in the future expand to frontotemporal dementia. Other assets focus on treatments for Alzheimer's disease and epilepsy syndromes.
The science behind that was actually quite, we didn't start out obviously to expect that this is, the discovery that we were gonna make. Everything happens. There is no eureka moment, so to speak. We had the idea back then when I first started my PhD to actually do a screen of one of the lead protein of that has just came out at that time to be identified to be implicated in ALS and frontotemporal dementia. And that's this protein called TDP-43. We had the idea to do a screen using bacteria. And that's where we identified the interaction partner, which was which would later on play a really key role in the disease. And it's from there that, all the work started. And that took, I don't know, 15, 16 years. And during this time, we did a lot of very detailed understanding into the role of this particular protein and identifying the regions that the interaction happened and how the interaction happened, when does it happen, and what does it do to the disease. And that kind of was the bulk of, the time that was spent. And then from there, we took that information that knowledge behind what we could understand in the disease mechanism, and from there developed this therapy.
So for me, a key observation that we made when we found the interaction between 1433 and TDP-43 was recognizing that there is a naturally occurring sequence that preferentially binds to pathological forms of TDP-43. And that really sparked the idea that we might have just come across a molecular tool to tag these pathological forms that appear to be the problem in neurons and leading eventually to their degeneration. And building on science by others as well as our own discoveries, we devised CTX1000 into a genetically delivered medicine that can specifically remove pathological forms of TDP-43 from neurons, and as such enable these neurons to recover at least in parts and take up then their function again. In the process, in the discovery process, one essential step is subjecting your research to the peer review process and get feedback from colleagues, and to improve the study and ensure that all findings are valid. And we were very pleased that why I really I think very positive process that we experienced with Neuron, that eventually this paper got accepted in this high-profile journal, and which was not only key to finally broadcasting our discoveries to the research community, but formed the basis for the interaction with investors and really forming Celosia to bring this, translate this into clinical trials and hopefully treatments for people.
So since the paper came out officially in February last year, so we had obviously gotten a lot of media attention because that is probably the first disease-modifying treatment that's available for patients with ALS. And in the scientific field, it's obviously hailed as one of the breakthroughs. And since then, I've gotten a lot of invites to talks, gotten a lot of invitations to comment on this. So I think the feedback has been overwhelming.
I think to me the key feedback beyond the media attention and the feedback from pharma and industry was personally that very senior, very experienced colleagues in the field have reached out and in conversations really appreciated the work we've done, acknowledged the amount of work this has gone into and the potential this has for therapy. So the next step for CTX1000 is the transition to clinical trials, which we aim to start in Q3 of this year. For that, a clinical protocol has developed, which we are doing with a very experienced research organization. So the first clinical trial here at Macquarie University will be a phase 1b trial. The primary objective of this trial is to test the safety and tolerability of CTX1000, with secondary endpoints being potential efficacy in slowing the progression of ALS.
So currently for CTX1000, we are developing the process that's needed to manufacture CTX1000 to a scale that is required for human trials. We are going first in human trials sometime this year, and for that we need a batch that is human ready. And at this point is developing the process with our CDMO, our manufacturing partner, and trying to understand how to develop that at a scale that is required and safe as well to be administered in humans.
So we have fortunately secured one of the internationally leading ALS clinicians as principal investigator for this study. And we are looking forward to this study commencing here at Macquarie University in Q3 of 2025, so this year. My hope for CTX 1000 is that 15 years of research where we've seen significant improvements in many different experimental models of ALS will translate into tangible outcomes for patients where we see significant slowing of disease progressions or even halting the disease progression altogether.